We will continue with some introductions, and then we will hear from our featured speaker, Dr. Gentry. He will be speaking on microbial source tracking analysis of recreational waters in Galveston, Texas. After that, we will move into a Q&A session, and then we will wrap up by noon. So again, we will be using that chat function for questions. Feel free to put your questions or comments in the chat at any time during the presentation, and we will then address them after the presentation. Also joining me from Meadows Center is Sarah Wingfield. Sarah is our digital media specialist, and she is our tech expert for the Lunch and Learn. So if you have any technical issues during this call, please feel free to send her a direct chat. Now I would like to introduce from the Texas General Land Office, Jason Pinchback. Jason?
Good morning. Thank you so much, Christina. Welcome to another installment of the Texas General Land Office Clean Coast Texas Lunch and Learn series, where we connect with coastal water resources and learn about ways to understand and enhance the management of stormwater runoff. My name is Jason Pinchback, the Water Resources Manager at the Texas General Land Office, working under the leadership of Commissioner Don Buckingham. This event is sponsored by the Texas Coastal Management Program, and it is intended to be an easy-to-access forum where we invite subject matter experts to unpack their initiatives into bite-sized morsels, to share ideas that might be useful for neighboring coastal communities on how to improve local water quality, habitat, and related resources to help support economies who thrive on clean coastal waters. Future Lunch and Learn events coming up next month in September on the 19th will look at how water quality impacts local economies. This will be presented by PhD candidate Virgie Greb. Please pardon our delay in that edition as we had interruptions due to both a bridge and barge incident as well as Hurricane Beryl. We'll be back next month. Clean Coast Texas is here to serve communities by providing local governments, decision makers, and the public with information, resources, and tools to better manage stormwater quality and its associated runoff. The tools and guidance provided through Clean Coast Texas can help coastal communities implement measures to reduce the impact of land use change on coastal waters and protect natural resources that help support thriving coastal economies. Some of these services include community planning and ordinance development, water quality analysis, and green infrastructure workshops that help to bring meaning and value for how to implement some alternative building and planning practices. We also work with our partners to develop grants and then manage those projects, and then do other things such as stormwater retrofits and green stormwater demonstration projects. And there's many other measures as well, including public engagement materials. We have a lot of that information available on our website, cleancoast.texas.gov. And I think we'll have a link to our menu of services in the chat box here in a little bit. To assist us with this initiative, and knowing that this type of challenge takes a team of well-seasoned and knowledgeable practitioners, the GLO formed a collaborative with the Meadows Center for Water and the Environment at Texas State University, with Texas Community Watershed Partners, AgriLife Extension Service, as well as Texas Sea Grant at Texas A&M. It takes a team. I would like to thank Sarah Wingfield and Christina Lopez at Meadows Center for Water and the Environment today for co-hosting and providing technical support for the Lunch and Learn events. I'd also like to take a moment out to acknowledge our very own summer intern at GLO, Miss Edith Holmston. She's been with us for a couple of months and be with us for the rest of this month. She's provided a huge lift in assisting the important Texas Beach Watch and water resource activities and initiatives. She is about to begin her junior year at University of Washington, Seattle, studying marine biology. So thank you, Edith, for your valuable contributions. Today's featured presentation is provided by Dr. Terry Gentry. Dr. Gentry is a professor of Soil and Aquatic Microbiology in the Department of Soil and Crop Sciences at Texas A&M University in College Station, Texas. His research focuses on applied microbial processes, including detection and remediation of soil and water contamination and sustainability of intensive cropping systems. He has co-authored or authored 112 peer-reviewed journal articles in addition to 15 book chapters and leading textbooks on environmental microbiology and soil microbiology. He has developed and instructed numerous courses, including soil and water biology, environmental microbiology, and environmental soil and water science. I would also like to add that he and his team at AgriLife have been top-notch partners who have been instrumental in helping us to better understand coastal water quality. So let's buckle up, put on your thinking caps, and get ready to learn. Without any further ado, let's welcome Dr. Terry Gentry. Terry, we see your screen and your presentation.
Great. Well, good morning, everybody. Glad to be with you and present. Howdy to any Aggies that are out there. And so today what we'll be doing is, as mentioned, is talking about microbial source tracking and some of the work we've done in Galveston. And this was done by a former master's student, Carolee Corbell. Carolee is now a molecular technician with the University of Texas in Austin. And of course, we have technical issues right out of the gate. There we go. So first off, what is microbial source tracking? So the idea behind this is looking at and trying to be able to determine sources of fecal contamination. If we have a water body and we have high levels of fecal indicator bacteria indicating there's fecal contamination, one of the most important things up front is try to determine where that's coming from in order to address the sources. And so if you look at the different pictures on the right, you got several different animals that are there, including humans. And the idea behind source tracking is the microorganisms that are in the intestines of those animals are different enough that we're able to differentiate them when we see them in a water sample. So you think about the intestines of humans, the intestines of those animals, there's going to be millions and billions of microorganisms represented by thousands of different species. And so there's enough of those that are unique in the different sources that we can tell them apart when we see them in a water sample. There's been a huge number of different methods that have been used, and we'll touch on a few of those in upcoming slides. But oftentimes, the source tracking works best as a toolbox approach. For example, if you need to drive a nail, a hammer is probably going to work better than a wrench. In contrast, if you're trying to tighten a nut on a screw, it's going to be easier to use a wrench. And so the same type of thing applies in terms of source tracking, that one method may be best in one situation, but wouldn't be best in a different situation. Now, I'm referring to microbial source tracking here. You may have heard the term bacterial source tracking, and I'll probably use that before the presentation is up. They're essentially referring to the same thing. Microbial source tracking is a little broader, recognizing there's other things besides bacteria that could be sources. And for example, if you look at some of the different targets that have been used, we have very common bacteria, things like E. coli, Bacteroides, we'll talk a little more about on an upcoming slide, and then viruses, things like bacteriophage. A lot of viruses that infect E. coli, for example, have been used or tried to be used for source tracking. There are human viruses, even animal cells. If you're familiar with environmental DNA or eDNA and its use in detecting rare species in aquatic systems, there's the same type of thing can be used in microbial source tracking to detect the amount of cattle or something else, for example, that could be interacting with the water. And then lastly, even chemicals. And a couple chemicals have been used, one caffeine, with the idea that human sources are probably one of the biggest for caffeine that we see in the environment. And then also optical brighteners are chemicals that are put in laundry detergents to make our whites whiter, because that can be an indication when we see it in the environment of sewage or septic, for example. But in this case, we're going to focus on the microbial topics or targets, and I'll kind of group all these different types of approaches into three categories. So the first of these being culture-based approaches. And so in this case, we isolate our bacteria, and then we'll try to characterize that using either a phenotypic or a genotypic approach. The phenotypic approach would be something like antibiotic resistance. The genotypic may be DNA fingerprinting or something like that. And then we compare those isolates back against ones from known sources. So the way this would work is we have a watershed, for example. We think that there's humans, cattle, and deer are our main sources. We would collect fecal samples from all those. We would isolate bacteria. We would do our DNA fingerprinting. Then we go to the water sample. We isolate the bacteria. We do the same fingerprinting, and we compare those. So it takes a lot of time. It can take a lot of money to build that known source library. With that said, that's been the core of the work we've done in Texas for the last couple of decades, and it's been used to do source tracking in multiple different watersheds across the state. Now, the second approach is marker-based. And this one and the next one are the two we're going to talk about being applied in this study. In this case, we'll take that water sample, we'll extract the DNA, and we'll use polymerase chain reaction to amplify specific DNA segments or targets. And then the third approach is a sequencing-based. In this case, we're going to extract the DNA as well. And instead of using polymerase chain reaction to amplify, though, we're going to do DNA sequencing. And then we're going to compare that back against the community's known source. So on the next couple of slides, we'll go a little deeper on those last two methods. So first off, with the marker-based MST, so the most common of these approaches target Bacteroidetes, not E. coli, not Enterococcus, not some indicators, but instead Bacteroidetes. These are very abundant in the intestines of animals. In fact, they're more abundant than E. coli in most intestinal samples. With that said, there are individual groups that are relatively specific for different types of animals, and that's enabled the development of markers that are specific for things like ruminants, humans, horses, birds, and hogs. Now, a challenge of the marker-based approach is that there aren't very many markers for wildlife, and most watersheds, most environmental water bodies are going to have some contributions from wildlife. So if that's a concern, that may be a limitation to using this type of approach. Also, the relationship between these markers, again, they're based on Bacteroidetes and some other organisms. They aren't based on our indicator organisms like E. coli, Enterococci, and aren't based on the pathogens. So that relationship between what we see in terms of the quantification of these markers and our regulatory organisms or even the pathogens themselves is a little bit uncertain. And then also, there tends to be a trade-off between specificity and sensitivity with these different markers. Is we have some, for example, human viruses. If you see that in the water body, that's a pretty good evidence that there's human contribution. But in most cases, those human viruses are only being shed by a relatively small portion of the community. And so you could have a lot of human contribution that just isn't being detected because the people that are shedding the fecal material don't have those viruses or aren't infected with them. And so you get this trade-off between false positive and false negative that will impact the methods that we use depending on what the goal is. But the way we apply it is take the water sample, extract the DNA, do quantitative polymerase chain reaction to amplify it. And this is based again on the fact that these organisms, they're specific enough, the DNA sequence of them are specific enough, ones in cows, for example, versus humans, versus gulls or something else, are different enough that we can detect those specific DNA segments in those samples. And then we can quantify that to get an abundance of it. Now, with a sequencing-based approach, this is a little different. Again, it's based on DNA, but in this case, we're going to sequence it. We're not using polymerase chain reaction to amplify different segments. So we'll extract the DNA, and we'll use high-throughput DNA sequencing, thanks to the rapid advances that have happened since the Human Genome Project and sequencing technology. This is becoming much more doable. And then we'll compare those microbiomes using computational programs. So if you look at the figure on the bottom, kind of the way this would work is we take our animal fecal sources, and so we do need to have an idea what we're looking at. So let's say we're working with cattle and deer or elk and cats in that example. We take the fecal material, we extract the DNA, we do high throughput DNA sequencing, and then we do the same thing with an environmental water sample. We extract the DNA, do high throughput DNA sequencing, and then we compare the two. Now, what this is doing in contrast to the marker analysis is with the marker analysis, we're looking for very specific organisms. We know up front what we're looking for. We're looking for Bacteroidetes with a sequence that's specific to cattle or specific to a different type of source. In this case, we're just looking at what's there, and then we're comparing after the fact how similar they are. And so this example, for instance, is we're taking a water sample and we're seeing how similar the bacterial community in that water sample is to that cat sample. How similar is it to that cow? How similar is it to that deer or elk? And so we're looking at a lot of different organisms. These are probably not pathogens. They're probably not our indicator organisms, but we're looking at a lot of different ones. So it gains some strength from looking at multiple different bacterial species. So let's look at some actual results from the work we did in Galveston. So this was work with Texas Beach Watch, and we're using the routine samples that they pulled every 1 to 2 weeks across Galveston. but we targeted the samples only with high enterococcus counts, so greater than 104 mpm per 100 mils. Throughout 2022 and 23, we collected 114 of those that were analyzed, and we used two different source tracking approaches. The first being that quantitative polymerase chain reaction marker analysis, and we looked for three different sources or three different markers: human marker, dog marker, and gull marker. The human and dog target Bacteroides. The gull targets a different bacterium. And then we also did microbiome-based BST, or the DNA sequencing BST. Again, we targeted the same sources: human, which included wastewater treatment facility and septic samples, dog and coyote, and then also gull. And again, that microbiome, the idea is we're comparing it versus those known sources. And if you have questions, feel free to add those in the chat, and we'll get to those after we get to the conclusions here in a few more slides. So let's look first overall at the marker results. And so this is the overall marker results. And it's laid out by the different months. So starting in March of 22 up through May of 23, the y-axis are the number of samples that were collected and analyzed. And the gray part of the bar are the samples that were negative for any of the markers or all the markers. And then the maroon part of the bars are the samples that were positive for at least one marker. And so looking across there, especially in 2022, you see that there's very little of the markers, very little of the samples that tested positive for one of the markers. Where we really started seeing it was in the 23 samples. And overall, about a quarter of the samples tested positive for at least one of the markers. But again, that was kind of stacked toward the latter end of the study. And so that's one of the things we'll come back to in a little while, is that in 2022 is a really good water quality year for the Galveston area. And so that probably has some type of impact on the results. In 23, we started seeing some of the marker abundance levels pick up a little bit. So next, let's look at each one of those individual markers. And so this map is going to have the same map on the next this slide and the next two slides. This is the map of Galveston. You can see the different sampling locations in maroon circles that are indicated, and these are the ones that tested positive for the human marker. There were sampling locations scattered across the entire island on the beach side, also some on the bay side, and even up into the shipping channel. But the ones that tested positive for the human markers are these 5 locations: 3, 5, 7, 32, and 47. And overall, that was 4% of the samples that we tested, 4% of the 114 samples tested positive for the human quantitative polymerase chain reaction marker. Now, in terms of levels, the highest level we saw was a little over 1,100 copies per 100 mils, and that was in that Site 32 sample from May 23rd, 23. And the reason to point this out is that, going back to what we talked about earlier, is there isn't a direct connection between these markers and our indicator organisms or pathogens. But one recent study has suggested a level of around 500 of this human marker per 100 mils would exceed acceptable health risk for recreational contact. And you can see in this case, we're well above that. So just based on what we saw in the marker here, just contributions from human sources would exceed what we would deem to be an acceptable health risk at this point. And so some indication of human contributions across the island during the sampling period. For the dog marker, relatively similar. And so see the red circles indicating the sites that tested positive for the dog marker. See a little wider distribution than we see with human samples, a little greater prevalence, 7% total of the samples that are here. But also there's an overlap. And so all the samples that were positive for human, 3, 5, 7, 32, and 47, also had a sample test positive for dog. And so it makes sense. There'd be a co-localization where you see dog and where you see human. But again, we did see some test positive for dog, and that was scattered across the island. And the gull marker. So this is indicated by kind of the burnt orange color, not intentional, but the orange color circles that are there. And you can see they're scattered across the entire island, both on the beach and the bayside. And around 24% of the samples tested positive for the gull marker. So by far the widest distribution, And we saw that in the greatest proportion of samples of the different markers that we tested. And if you've been to the beach at Galveston or you've been to beaches, there's gulls there. There's gull waste really in close proximity to the water. So probably nothing real surprising in terms of that. Okay. So how about the DNA sequencing or the microbiome microbial source tracking? So again, this is different than the previous analysis. In the previous analysis, we're looking for very specific organisms and are they there and in what abundance. In this case, we're looking at what is there and we're comparing it back against those different sources. So we kept the same sources. If you look at the legend on the right, we had the gull, we had the human, the wastewater treatment, and the septic. We had the dog and the cavity. So we're keeping the same general categories. Now, the way this is laid out is you look from left to right. On the left is kind of the west end of the island, then the seawall, and on the right's the east end. So kind of laid out as it would be geographically across the island. The y-axis is the percent source contribution. So what this essentially means is when you look at that water sample, what is the similarity of the bacterial community in that water sample, each of those different sources? Again, many of those are not going to be pathogens indicators. It's just going to be whatever bacteria are in that. Okay. So let's start looking at our different sources. So looking at the legend, the top one's blue. And across all those bars, in most cases, the largest portion of the bacterial community is being classified as unknown. Now, what does that mean? There could be a couple explanations for that. One, it could be a source. It could be a fecal source that wasn't human, it wasn't dog or coyote, and it wasn't gull, so we didn't see it. Or it could be just naturally occurring bacteria that you're going to see in aquatic systems and just part of the normal microflora. And in fact, when we went in there and we started looking at the names of these organisms and who they were, many of them were cyanobacteria and things you would expect to see in a very natural, normal water body. And so that's probably what the vast majority of those, that blue part of the bars are. So let's focus in on the ones that were actually very similar, or the portion that was similar to the sources we looked at, the fecal sources. So the ones we tested, the one that was largest is that teal colored part, which is the gull. This kind of matches up to what we saw with the marker analysis, with the gull being the largest in proportion and the widest detection of that across the island. So especially on the west end, on the left side of this graph, you see relatively good similarity between the water samples that were pulled as part of Texas Beach Watch and the gull fecal analysis. Now, again, there can be a couple of reasons for that as we're looking at similarity. Is one, the gulls live there, they're eating things that are in the water, they're in contact with the water, so you'd expect to see some similarity between the water and their intestinal community. But there's definitely gull waste that's present. You can see it easily. So if we look at the next highest level, that would be that lighter green color, which is wastewater treatment facility effluent. This is the treated effluent as it's being discharged in the outfall. So this would be expected to have much lower levels of indicator organisms, lower levels of pathogens. But we are seeing a relatively good similarity between the bacterial community that's coming out of that effluent from the wastewater treatment facility and what we see in those beach watch samples. And then how about the other ones? Coyote, human, dog don't see a whole lot. So you look at those orange, red color parts of the bars. There's a little bit, you can start to see a few little segments at the bottom in a few cases, but outside of one sample, we saw a few percentage of coyote. For humans, it was relatively small proportion. About 2/3 of the samples appeared to have human contribution, or at least similarity, less than 1%. Now, with that said, the human samples may have the greatest risk just because there's much more likely to be carrying human pathogens, viruses, and things that the other sources wouldn't. And so even though there may be a low level in terms of similarity, that doesn't mean that there isn't any potential risk from contributions from even a 1% human sewage source. But overall, pretty good similarity between the results we saw here with the microbiome MST and then the marker analysis. Now, one thing to point out is that these MST analysis was just in the 2022 samples. And again, that was the really better water quality, so that probably had an impact on it. So overall, what can we conclude from this? Is that with this preliminary analysis, And again, these were relatively limited number of samples. We're talking about 114 samples, primarily over 1 year, spilled it a little bit into the next year. But it was likely impacted by both the study design and that really good water quality in 2022. With a study design, these were using existing sampling locations on a weekly, maybe bi-weekly basis. And so if we went back in, we did more targeted sampling, we may see a little different results. With that said, we did see markers present for human contributions, dogs and gulls. And then the source tracker analysis tended to corroborate this or help verify it. Also, the source tracker analysis indicated a potential impact of that treated wastewater treatment facility outfall on the aquatic microbiome. Now, whether this is contributing indicator organisms, whether this is contributing pathogens, that's a different question. But there appears to be a similarity there, or there is a similarity, and that appears to be indicating some type of potential impact. And then, as an academic, we always got to say more work is needed. And so, if we did additional analysis, if we had especially more analysis in a year where the water quality wasn't quite as good in terms of fecal indicator bacteria, we may see better results in terms of where those sources are coming from. And then also doing targeted sampling is rather than doing the beach wet samples, which are going out kind of at knee depth level and pulling a sample, is we get closer to potential sources, outfalls, for example, where there's less dilution. It may help us in getting better detection of some of those markers. And the last thing is we're doing ongoing work with Anna Gitter at UT School of Public Health in El Paso using quantitative microbial risk assessment. And we're doing this with that existing data that Carolee collected. And the idea behind this is we're talking about these different levels of the human marker, for example, versus the dog versus the gull. How does that translate into actual health risk? And so we can go back and we can use historical data, literature values for different pathogens that are present in those different sources, and then try to put this into an actual number. We're saying quantitatively, if we have 1% of human, does that actually exceed 30% of gull, for example? So we're trying to put it back in terms of an actual number for human health risk. And with that, I'd just like to acknowledge that the project and the work was funded by the Texas General Land Office with help from Jason, and really appreciate Lucy Flores and her crew for collecting the samples and making those available to us. And this project was done in connection with Texas A&M AgriLife Extension with Anish Jantrania and Gabriele Bonatti, who a few of you may have heard present a few months ago. And with that, I would be glad to try to answer questions now. So here's my contact information. Feel free to contact me later if you'd like.
Thank you so much, Dr. Gentry. That was a great presentation. You added a lot of information in a tight period of time. And I even understood half of what you said, I think. So please, folks, we do have some questions in chat. If you have more questions to add, please put them in the chat and we'll be facilitating those here momentarily. I'll get us kicked off, Terry, with a question about viability of what we're testing for, viability for detection. So we have different inputs from the different animal sources. So seagulls, dogs, coyotes, etc., are depositing directly onto a nearby surface. Whereas hopefully the human sources are not being deposited directly, but are probably taking alternative pathways, such as moving through a sanitary conveyance system, underground, overground, and then eventually making its way. So the time before it makes its way to the water is going to be different for all these different species. Can you comment on just the viability for detection from the different species, if they're different, if they're all the same, and how that impacts results?
No, absolutely. And that's one of the things that really complicates it, especially if we, excuse me, start looking at different organisms. So, for example, some of the Bacteroidetes markers may die off relatively quickly, but some other markers may persist a lot longer. So it's kind of the same thing you think of like with using E. coli versus enterococci. Is that E. coli is going to die off a lot faster in that marine system than enterococci will. Same type of thing happens with these other organisms. Some will last longer, some are going to die off much more quickly. And so it absolutely complicates the analysis on it. And then if you start factoring that, like you say, the spatial and temporal differences in terms of where they're deposited, the amount of time it takes to get into that water, we're detecting it, that just throws a whole other level of complication into it. And so that's where it's always good to have multiple lines of evidence to help figure out what really is happening there. Is that we can go in there, we can measure it and think we got it nailed in terms of exactly what's happening. And unless we consider those other aspects that could be impacting it, we may get a misleading understanding of the situation. So it's really important to consider all that.
Yeah, the results were really interesting as a part of this study. And of course, our design was built on the Texas Beach Watch monitoring schedule and protocol. And as you mentioned, the water data itself was relatively wonderful for 2022. So it's kind of like if you do a flood study, we go into a drought. Or if you're doing a drought study, then you enter a wet season. It's kind of typical of folks who do field work. But well, thank you for that. And I'd like to turn it over to Christina to help us facilitate questions.
Thank you. Great presentation, Dr. Gentry. We do have questions coming in the chat. If you would like to ask, please do not hesitate. So this is a little bit similar to Jason's question, talking about other species. We had two questions come in, one regarding cattle in the current study that you presented, and that they were maybe not tested for. So there's the cattle question, and similar to that, could you potentially test for monkey sources? Keith says there is a large monkey farmed plant about a half-mile from the San Bernard River. So you talk about cattle in this current study and monkey for future studies.
So I'll start with monkey, because I've been doing this for 20 years in Texas. This is the first time I've ever heard that come up, monkey. So that's a new one. I've heard a lot of new ones. So the short answer is no, we haven't tested for monkey. I don't know if there's a marker available for monkey, but if that's the case, and that's a very important thing, is normally when you go into this, and let's say we're doing it for a watershed with a stakeholder group, is that's one of the first things we want to do is figure out from them what they think the sources are. And if there's anything like that that we're not aware of, that we want to try to test for it if we can. But the short answer is I'm not aware of a monkey marker, and I haven't done any of that. But that would be a really good point. That could be a source. Going to cattle. There are cattle on the island. We didn't consider that as one of the major 3 potential sources. Again, this is preliminary. This is the first source tracking we had done in Galveston. And so we try to focus on the first 3, and those 3 are the ones that have been pretty commonly used on different beaches around the US. And so we prioritize those. Cattle wouldn't have been detected with any of the marker analysis we did. We might have seen some cattle with a source tracker, but we didn't include a known source comparison. And so when you go back to that blue bar on that source tracker graph that had the microbiome BST, some of the blue could have been cattle. We don't know the answer to that. But good question.
Thank you. This next question, I believe, is referring to the bar graph you just mentioned. They ask, what is the difference between the wastewater effluent and human on the side of the total contribution with all of the sources from west to east?
Okay. And so we're looking, y'all can see the bar chart now? Okay. Yes. So the question is about human going across the island.
The question is, what is the difference between the wastewater effluent and human?
Okay. And so the way it's labeled there is that green wastewater treatment facility effluent. That's what's coming out of the wastewater treatment facility. So that is treated wastewater. The other things that are listed as human, both wastewater treatment facility human and human septic, those are raw. They're untreated. And so that's the difference between those. And so the lighter green that we do see quite a bit of similarity, that's with the treated. And so that may or may not have pathogens, high levels of pathogens. You would hope it doesn't because it's been treated. But the red and the orange would. We didn't see too much of that. And so you can see this with right above the seawall area. You can see a little bit of red right above sample 25. But generally speaking, it is less than 1% similarity to raw wastewater. And we didn't see a really good pattern, if that's part of the question. As we look across the island, we didn't see a really good pattern in that. It was so low. And again, this was probably heavily impacted by that really good water quality. If we went back in a year where we saw a lot of elevated enterococcus numbers, we might see higher levels, or we expect to see higher levels, and it may give us some patterns. But in this case, those levels are so low, there's really wasn't a good pattern.
Thank you for clarifying. So staying on that same slide, the next question is, what do you suspect is contained in the unknown category, as it seems to be a large part of the samples? I know you mentioned potentially cattle.
Yeah, so absolutely. So two very possible explanations, and it's probably a combination of these, is one, these are just normal aquatic microorganisms, cyanobacteria, whatever, that you would see in a system with zero fecal contributions. And in fact, when we go back and we started looking at the ID of these based on the sequence, that's what a big portion of them appeared to be, is things like cyanobacteria. However, that previous question that asked about cattle, that's another potential source, is whether it's cattle, whether it's monkeys, whether it's anything else. If it's something that we didn't explicitly include in the analysis, that could be showing up as the unknown. And so that's always a question. And so it's really important up front to include as many sources as you can within budget to try to ascertain that. But it's a combination of those two things most likely. But I think the majority of it is normal aquatic organisms based on what we saw with the IDs.
Okay, thank you. Next question asks, are you using high throughput DNA sequencing to look for host species DNA, for example, human or dog?
We aren't looking for host organism DNA. We're just focusing on, in this case, the bacteria that are present within, so be shed by the animals, but we're not looking at the animal source DNA either itself.
Thank you. Next question is, aside from working towards quantifying local markers at the site, did you look into the influence of land use factors and water quality?
So in this project, we did not. And so it's more of a survey over time. And again, it was preliminary. And so we were focusing and prioritizing on the samples that were high to start with in order to make the best use of the funds and the resources, rather than just going in there and doing this on everything and ended up with a lot of non-detects. Is we tried to emphasize it on the samples that had high fecal indicator bacterial counts. But the accompanying project that with Gabriele Bonatti that he talked about last time, they did get into some of this and they looked at correlations and other ones. With my part of it, we didn't go into that part. But in terms of part of the bigger project, there was the look at it in terms of land use and the different potential sources that are within there.
Great, thank you. All right, next question. What is the difference between microbiome MST and the host-specific MST markers you used?
Okay, and so I may not answer this question the way it was intended, so feel free to follow up on it. So with the microbiome MST is we're sequencing the entire bacterial community. We're using 16S ribosomal RNA primers, general type primers, and we're sequencing that community. We'll, for each sample, get thousands of different reads back, and then we'll compare what's in a human sample, for example, with what's in the water sample. For the marker MST, would you refresh my memory on exactly how that was worded? It was the marker MST.
Yeah, so the question is, what is the difference between microbiome MST and the host-specific MST?
Okay. And so with the microbiome, that's representing the microbial community, everything that's there. With the host specific, that's generally looking at a very specific group of bacteria. So it could be a specific genus, it could be a specific family or class, but it's looking at a subset of that microbiome that tends to be specific in a general animal host. And so, for example, with the microbiome, we're going to have Pseudomonas, we're going to have Bacteroidetes, we're going to have all sorts of different organisms that are present. With the host specific, we may be looking at just the Bacteroides. It could be as specific as a species. And then within that, just the ones that are found in humans or just the ones that are found in a different source. So it's very general versus very specific.
Thank you for clarifying. This next question and potentially last question, so if you have a question, be sure to get that in. This might also be for Jason and Beachwatch. So the question reads, a fisherman died from Brivio in June in Freeport. Can tests easily be done and shared with the public when levels of Vibrio are high?
So Jason, do you want to get that?
Well, the testing for Vibrio, there's been a lot of interest in that, especially in the last couple of months, as there have been some reported cases in this part of the coast that we're discussing here. So at the General Land Office, we're currently not testing for Vibrio, and to the best of my knowledge, testing usually occurs after a case is detected, where follow-up testing occurs in that particular area to make sure that there's not some type of an outbreak or a larger event. Vibrio and the various forms of that bacteria are naturally occurring in many different parts of all the marine waters. And but scientists have not found that there's a strong relationship with many other or any other parameters to monitor. So I'm not aware of it being routinely tested, at least in ambient waters. And Terry, can you comment on Vibrio in general, or if there's any relationship to the work that you're doing?
So I would tend to go the same direction you are in terms of agreeing with that. It's there. It's just part of the aquatic microbiome. There may be things that we do that impacts its abundance, but it's just part of the natural community. And unfortunately, when there is an infection, it tends to be very serious. But in terms of the monitoring of it, that's not something my lab has routinely done. There are groups that can do that, but I'm not aware of it being routinely measured in the Gulf Coast area.
Thank you both. Next and potentially last question says to Dr. Gentry, you mentioned good water quality in reference to the 2022 PCR marker samples. Was this in reference to the lower percentage of markers identified, or was there some other meaning implied there?
It was based on the fecal indicator bacteria. So the enterococci levels is the samples we used was anything that exceeded 104 mpn. And there were relatively few that exceeded that in 2022. So it's based on the regulatory organism. This is the reason for that statement. In fact, we planned to just do this study in 2022, but we got so few samples, we extended over into 23 to get more. But that statement comment was based on the fecal indicator levels.
Got it. Thank you. All right. Well, that's all we have from the chat.
Well, thank you so much, Christina, for helping us with all parts of our Lunch and Learn today. And just if I could comment on the results here in the study, and I think you had it in your conclusion slide, Dr. Gentry, about these being preliminary. And this was our first attempt here, kind of getting our feet wet, so to speak, on looking at this particular study. We are continuing to adapt, to remain agile, and to refine the studying approach. And we are searching for funds right now so that we can enhance the money we're bringing to direct into this, and then treat areas that need to be treated and focus in on those. Dr. Kuknor did make a comment, and she is the lead of the Texas Beach Watch Lab out of Lamar University. And so she had a comment that her students have finished analyzing abundance of Vibrio for the last year, and they'll have results shortly. So we'll reach out and see if she's interested in sharing those results when they're ready, and maybe we can come back and share that in a future Lunch and Learn. Dr. Gentry, thank you so much for sharing your time and expertise with us here today. We really do appreciate that. And thank you for everybody attending here all the way to the very end. And we will see you next month for Virgie Grubs. Thank you so much.